primer, very brief primer on the virtual fair.
Okay, everyone. Yes, I want to thank all the presenters because they really worked really hard. Were really patient with me on booth setup. So great job, everyone. All right, let me share my screen and I'll walk you briefly through the campus group format. Okay, I'm hoping everyone can see this. Okay, so when you log into campus groups, you should be on this event page. And so you'll see the great picture here, registration here. And then if you scroll down under details, it will have the links for the day. So we are in the opening session right now. Then we will move to poster sessions. This is the actual fair with all the booths. And then at 3:15, we will return to the main session, this one here, where we will have closing remarks. Now, you can navigate a couple of ways. You can click this link directly here to go to the fair, to the virtual booths, or you can click this meeting link. So if you click on the meeting link, you will see the presenters. So these are the poster presenters. This is the list view. You can also go to the map view. And on the map view, you'll notice they're color coded. All blues are first poster session. The second poster session is in reddish. And then the closing ceremony is right here with Dr. Usman. So when you click on a bubble or a list, you will see the list of presenters under that category. If you click on their name, you will get all the information here. This is their Zoom meeting, passcode embedded, so you will not need to enter a passcode. And then you will see their pictures. Their document is their actual poster, so you can open it up for viewing. And this is their time frame. So they will be in their Zoom meeting during this time. You can enter and exit a booth as many times as you'd like, stay there as long as you want, but we would like you to visit as many booths as you can, if not all of them. And then if you just scroll down, you'll have more information if they included additional links for their presentation. Let's see. And then if you go back to the agenda just really quick, you'll see the conference link here. If you click on that, it takes you back to the actual event. Sounds good, everyone? Any questions? We're good? Yes. Okay, enjoy the conference.
Great, great, great. All right, well, let me ask, is Dr. Blanchard here? Yeah, I think he's not able to make it, and we didn't receive a video recorded message, so we'll just move forward. All right, let's, I'm going to introduce Dr. Mian Jang. He'll be our introducer, if you will, for the STEM keynote. And Dr. Jang, you know, I've known him since I've been here 12 years, and he's never said no to me. I'm just amazed at all the things that I request, and he's just such a team supporter in the best sense of that word. He's a associate chemistry professor, and he has been a faculty mentor probably before my time, but definitely all the years that I have been here. And what I appreciate most about him is that, you know, he has made ways of introducing our students to faculty and researchers in the medical center. And he's had many publications as well as conference presentations. And many of his students have gone on for master's or PhD. So it's just my pleasure to introduce Dr. Mian Zhang. And I'm going to change the view, Dr. Zhang.
Okay, so thank you, Dr. Parker, for the nice notes. Actually, I didn't do that much, and I'm just one of our fine colleagues. So I'm fortunate to have been working with UHD for this many years, because a lot of support for both the faculty, admin, and especially the students. So good morning, everyone. Again, I'm very pleased to introduce one of our own, used to be Dr. Dada Chang, as a keynote speaker to all of us. I was very fortunate to actually work with Dr. Dada Chang back to more than 10 years ago. When he took my general chemistry class, he started the first semester at the UHD. And then just during that first semester, he approached me for research opportunities. I have been working with like more than 200 students on one-on-one basis for undergraduates at the UHD. That's my fortune. But I would say that Mr. Dr. Chang, that he was one of like a few students who actually took initiative to approach to faculty for this. Usually we put the flyer for Welch Grant Foundation for the Scholars Academy associated programs for the grant sponsored program. But he was a few students actually approached the faculties, and then so really that surprised me. Then of course I'm very happy to accept him, and he did a wonderful job just during that first semesters, and then started with like electrochemistry to make sensors. I think trans history at the UHD really well presented for a lot of achieving students here, and a lot of us here. I mean, as a student can really follow the path. So he came from a very hardworking immigrant families, and he didn't have that privilege to take a lot of elite schools, go to, outside of Texas. And he just very down to earth, just chose us. Of course, we have been very proud of for UHD being one of the most affordable universities in the state of Texas. But we do not compromise with our vigorous academic program, by the way. So he did a wonderful job in the research with me, and he moved forward to work with Dr. Jules, who is an organic chemist. And then eventually he ended up with Dr. Jerry Johnson, who carried a lot of biochemistry research. So in his 3 or 4 years at the UHD, he already experienced 3 and more professors just within one department. And he got such an exposure, and that really made him the resume is a glowing resume. As just before, as Mr. Mercedes showed one photos, and then he was one of the presenters of the off-campus meeting, right? So really made him very competitive, stand above the average, and he, of course, he was accepted by Texas A&M University, and he's going to tell you a lot of stories about his time at A&M. And he received his PhD at A&M, and he spent 18 months as a postdoc at another College Station-based companies. Now he currently works as a senior scientist at the slogan companies at the UHD, not at the UHD, at Houston. And as he already said, and he put it in his CV, and he's very enthusiastic about like a global warming and climate change, a lot of justified cause. And he is down to earth person too. He put these very good ambitions into his daily work. So we are going to hear his story for the whole things. So I do not know if I miss anything that, and you can definitely add a lot of details. I just want to say thank you and welcome back. And then a lot of students now will be very fortunate, since you are working locally, then you probably have a more accessibility to all of us. So welcome. So it's your floor now.
Thank you, Dr. Zhang, for the really nice introduction. Brought me back to the first day when I first came to talk to you and asked you to join and do research in your lab. It was 10 years ago. It's crazy how fast time flies. So Yeah, so I'm gonna share my screen now. Let's see. Can you guys all see that?
Yeah.
All right, so
Yes.
today I'm really excited to be invited to be the keynote speaker for the Student Research Conference at UHD today. So when I got the email from Dr. Parker and she said, you will have 15 minutes to talk about your undergraduate experience and then your PhD journey and my career in the industry. And I thought to myself, how in the world I'm going to talk about myself for 15 minutes, right? And then when I started to put all the slides together, I was like, oh, wow, I need to cut a lot of slides out because I didn't have enough time to actually talk about every single thing. So hopefully today I will, share my experience, undergraduate research experience at UHD, and we'll talk about my graduate school journey and how I transition all that to working in the industry right now. So hopefully you guys can learn something about me today. So a little background about myself before we dive into my journey. So I came to the US from Vietnam in 2006. So I graduated high school in 2007, and then I didn't know what I wanted to do after graduation. All my friends, they already know that they want to become a doctor, they want to be going to medical school, they wanted to do business school. I had no clue what I wanted to do. And that's okay, right? Because that part of life, you're still young, you want to figure out what you wanted to do. So I took some classes at Houston Community College just to basically testing out the waters, take as many classes as I could, basically just to learn about myself, to figure out what I am good at and what I wanted to do, for the rest of my life. So after getting my associate degree from Houston Community College, I joined University of Houston downtown in the fall of 2011 under the scholarship of Scholars Academy. I didn't know back then that was the biggest decision of my life that gonna change my career path, my life. But now I know, looking back at it, that was one of the best decisions that I made. So let's see. Okay. So the research experience at UHD, as Dr. Mian John mentioned during my introduction. So I did one semester in Dr. Mian John lab, working in electrochemistry. And after that, I moved on to work several semesters in Dr. Robin Joe's lab in organic and polymer chemistry. And before that, I had no idea what research was. All I heard about research was from the textbook, right? You heard a lot about, you form an hypothesis, you come up with an experiment, you do other experiment to prove or to disprove your hypothesis, and all of that before joining, the lab with the professor. I didn't know what exactly is the research means, but when I, joined and doing undergraduate research in Dr. Mian Jan and Dr. Robin Joe's labs, I actually got exposed to what science and doing research really are. I had hands-on experience. You come up with the hypothesis, you actually design the experiment, you actually do the experiment, and you analyze the data. And using all of that, you have to go back to your hypothesis and then prove or disprove your hypothesis, right? So all of that makes a lot more sense now, just compared with what you read from the textbook. So during my time doing the undergraduate research, I had so many opportunities to attend and present my research at both UHD Student Research Conference and national conferences. So the pictures that Dr. Parker showed earlier was actually from SACNAC 2012 in Seattle. And this is the group that some of them you can recognize, they in that pictures. So it was really funny when I look at that picture. I said, wow, it was a long time ago. And looking back, it's like, I think to myself, I couldn't make it this far, But yeah, so doing research in the lab is different, right? You learn about a problem, but to present a poster or giving a talk at a conference is another level. You actually have to dig really deep to understand the basic problem, and formulate the question and try to explain that to the audience, right? So that it's just not just doing research is to, understanding what you actually doing. That's the more important thing about, attending and giving a talk at conferences. So I had a lot of fun. Yeah, and I enjoyed it very much. So working with Dr. Minjan and Dr. Joes, actually, those two professors are brilliant. They actually influenced me and motivated me to pursue a higher education in science. And with the experience that I got, doing research at UHD, it actually helped me to get into graduate school, because when you went through the interview process, all they care about is what you did during your undergrad, what research you worked on, where you be able to explain it to the interviewers and convince them that you are qualified to actually join graduate school to do more research, right? So the experience I got from UHD that actually really helped me to get into graduate school, and I'm always grateful for that. So let's dive into my PhD journey. So I joined Texas A&M University, the Department of Biochemistry and Biophysics in the fall of 2014. I know about this department basically from a field trip, of Scholars Academy. This is the early day when I first joined University of Houston downtown. I think the field trip that I took was like early 2012. And that was the very first school that I visited. And I got the very nice impression from the Department of Biochemistry and Biophysics. And I was like, I'm going to go here no matter what. If I decided to go to grad school, this is where I wanted to be. And yeah, I applied and get into the program, and I just went for it, right? But the PhD journey is going to be different from people to people. If you ask the people who are actually working on their PhD now, or the people who already got their PhD, their story is going to be different, right? For me personally, I think the most challenge here is my first year. You went straight to graduate school after your undergrad, right? And during your first year, you had to do different lab rotations. So you have to talk to the professors. You have to prove that you are qualified, you're a good fit for the lab, because after that lab rotation, you have to pick a lab to join permanently, and that lab will be your home for the next 4, 5, 6 years, depends on how long your journey lasts, right? So that was really challenging. But on top of that, you doing the rotation while you're taking the graduate level classes, right? You want to make sure that you didn't fail any classes, while you have to prove to the PI that you're doing the lab rotation and that you are a good fit for the lab. So there was a lot going on in my first year. And on top of that, molecular biology was a big challenge for me. My background is in chemistry. As I say earlier, I did research in electrochemistry and organic and polymer chemistry. So molecular biology is just like a new concept to me during my first year, and it took me a lot of time to actually master different concepts and even the classes that I was taking during my first year. So that was another big challenge for me. So then, you survive all the classes, you survive the rotation. By the end of the first year, you have the exam you had to take in order to move forward to the second year. So they call it the original research proposal, so ORP. So this one, you had to come up with your own research idea that no one else in the department was currently working on. So you had to go to the literature, you had to read about so many papers, and you had to come up with an idea to formulate that idea, and then you have to propose different experiments. Again, come up with your hypothesis, propose the experiments, and then basically this is your child for several months. And you have to come up to all those ideas, and you have to write it, you have to defend it in front of a committee. Right. So that was really, really tough. And then, as a first year student, you heard so many horrible stories from, senior students saying that that's the worst exam ever. It's going to be so stressful. If you didn't pass, they're going to kick you out and all of that. So I prepared my original research proposal. I went into the exam room as, I was, feeling very confident as, first year student already. We were really passionate, really passionate about everything. And then I failed the exam the first time. right? That was basically a slap in the face for me to say, okay, I prepared everything that I could come up with, and then I still failed the exam, right? But and I was thinking to myself that is it, they're gonna kick me out. This is the end of my PhD journey, after the first year, and this is it. But they gave me a second chance and say I took it, right? So this is more important. So you took the second chance, you learn from your mistake, you learn from your failure, and then you retake the exam. And then I passed the second time, right? So I passed it and then move on to the second year. So again, just during my PhD journey, I enjoy it, but the first year I didn't enjoy it very much. So to move on to my second year, I joined Dr. Margaret Glasner lab in spring of 2015 to study enzyme evolution and mechanistic enzymology. So during my time here in Dr. Glasner lab, working on and study enzyme evolution and enzymology, I had so many opportunities to present at regional, national, and even international conferences to showcase my research, right? So at those conferences, you had a chance to meet with other fellow graduate students all over the places, and you had a chance to talk to professors. So those professors, they are potential employers, right? So if you just randomly talk to them, you never know, they might be hiring. So just want to point out that networking is very important, as Dr. Parker always say, right? If you go to a conference, just to talk to people randomly, you never know if they actually looking for a student in the lab or you're looking for an employee. Just if you talk to them, you never know, right? So again, networking is really important, guys. So keep that in mind. So this is several pictures that of different conferences that I went to throughout the year. One of the ones that stood out the most is that the Golden Research Conference in 2018. This is the international conference that happened in the US. So I had a really great opportunity to meet with a lot of people from all over the world and still in touch with them to this day, so it's pretty great. So, for PhD journey, it's not just doing research, right? It's not just staying in the lab and doing experiment. It's with, other things going on during the PhD journey too, including teaching and mentoring experience that I got from Texas A&M. So during year 2 and after that, I had an opportunity to teach for the graduate level and non-major biochemistry 1 and 2 courses. It was a really fun experience. I enjoyed it very much, but it could be a challenge because if you were teaching when you had a lot going on in your lab, or you did deadline, or all of that, it could be a challenge, right? So balancing between teaching and doing research, working in the lab could be a challenge, but that's something that you learn along the way. So one of the highlights that I got from my teaching experience is to get in teaching award from the Biochemistry Graduates Association in 2018. So I was really proud of it. I got nominated and I didn't know that I was going to win, but I won it. So that was really a highlight for me. So I really enjoyed teaching. Besides teaching, also mentoring is another experience I got during my PhD journey. I had an opportunity to mentor the first year graduate students, to help them, to guide them, for them to get through the first year. So because many of them will find the first year to be the most challenging, right? Like I did. So I've been there, done that. So to help them, to guide them get through the first year was a really honor, a great experience too. And then to mentor the undergraduate research, right? So to work in the Glasner lab throughout the years, we had so many undergraduate students coming in and they wanted to do research, as I wanted to do research back in my undergraduate at UHD. So I had the opportunity to, design, to help them design experiment, to help them with the experiment, to actually teach them a different aspect about research and mentoring. And that was a really great great experience that I got during my PhD journey too. And some of them move on to go to graduate school or medical school. So that was great. So in year 3, this is a really critical year. So you had to pass a prelim exam to become a PhD candidate, right? This is crucial because if you fail the exam, basically you're done, right? So the prelim exams consists of the written part and an oral part. So for the written exam, I enjoy it very much. So each of the committee member give you a topic, and you had to basically write out whatever you can come up with to answer the question. Right? I remember clearly one of the committee member just on the title of for my exam, he just put glucose, like one word. So what you do, you go to literature, you a bunch of papers, all the papers that you can read and write everything that you read about one single molecule glucose, right? So all of you who taken biochemistry too with Dr. Jerry Johnson, you know how important glucose is. So I enjoyed the reading part very much. For the oral part, it's a little bit tough because you had to stand up in front of the 4 members, and then you have to propose your research, all of that. But overall, it was a fun experience. I passed it to become a PhD candidate. This is like back in 2016. So you spend the next 2-3 years, working on, after you get approved your proposal, you spend the next 2-3 years to finish up all the experiment that you propose. And about year, 5.5 or so, now this is the time that you have to prepare to defend, for your dissertation. This is like the big moment too, right? So once you get a green light from your committee, say, okay, you are ready to write, you are ready to defend. This is it. So yeah, I spent months writing like almost 200 or 300 pages of my dissertation, and then prepare for the oral defense. Basically, you go in there, you just like pour your heart out and defend it, right? And I passed. So I got my PhD. Again, it was tough, right? But it was a fun experience. So my graduation is in May of 2020. And again, all we know is pandemic. So it got canceled. So I didn't have a chance to actually walk the stage, but at least I got my name flashed out in the screen of the Kyle Field, which is the biggest stadium in Texas, which is really nice, even though it's for like 1 second, right? So after my PhD, I got my PhD, now what, right? So you have to hunt for jobs. Jobs here, for a PhD path, it's not just like you get your PhD and you go out there and you get a job right away, and they make you to go, through postdoctoral training, right? You need to spend couple more years to get more experience. So for me personally, I asked myself, okay, so this is the crucial time. Is it academia or is it industry, right? Because this point, whatever lab, whatever path I choose, gonna determine the rest of my life, right? So I had the opportunity. Well, I got an offer from Chris Whitman, Texas A&M University, UT in Austin, and also another offer from Bondwell Technologies. It's a startup company that resided in College Station. So both offers were really, really good, but I decided to go with Bondwell Technologies. And there's many reasons why I picked Bondwell Technologies. So again, Bondwell is a small startup company located in College Station. So I joined in April 2020, like a month before my graduation due to the pandemic. And I spent 18 months there working in a really, really fun project and really exciting too. I had an opportunity to work with a variety of enzymes, come up with new enzymatic assays and development. It was really, really fun. And the project was funded by NASA. So by the time that it was looking for enzymologists, I would just like graduated, and it was just the right timing. And is in the picture here is Dr. Bondo. She was the co-founder the company, and I met her actually at one of the conference that I went to. And again, I brought up the importance of networking, right? You never know who you're going to bump into. So to talk to people, and this is sort of Bondos. She knew about my research. She knew about my reputations, I guess. And then I got a job to work for a really fun company and really fun project, and forever grateful for that. So for a small startup biotech company, there's several challenges, right? It's small, early stage, so there's a limited resources and funding, and some enzymes that I was working on, they were really difficult to work with. They didn't really want to collaborate. Yeah, but after 18 months at Bonwells, I got getting more experience, with enzymology, developed some of the enzymatic assays I never worked with before during my PhD, playing with some new instruments I never played with before, and learn a lot more about protein chemistry. And I, use all of that experience to move on to where I am at right now at Solugen, which is residing in Houston, Texas. So I moved back to Houston to take on this job. Right now, I'm working as a senior scientist in the enzymes group, working with other enzymologists and enzyme engineers. So the core value of Solugen is to fight climate change by going carbon negative, right? Because global warming and climate change is a big issue right now, and this is one of the many reasons why I wanted to join Solugen. Besides the fact that it's in Houston, so it's time to move back to Houston. Yeah, so we use green chemistry with enzyme, a main start, to produce chemicals. Yeah, so I've been with Solugen for almost 6 months now, and so far I've been enjoying every day working with enzyme very much. So it's really fitting to bring this today, because today's Earth Day, right? April 22nd. So it's real, guy. Climate change is real. Global warming is real. So if you're really interested in what we're doing, what Solugen is about, we are hiring. So check out our website and to learn more about what we're doing. Solugen is not paying me to promote Solugen right now, so I'm going to leave it there, right? So I leave the link so you guys can go check it out. So to conclude my talk today, again, the research experience I got at UHD really motivated me, really influenced me to earn my PhD and pursue a career in science, right? Working with Dr. Mian Jan, with Robin Joes, they are really influenced me with my career path, and I'm forever grateful for that. As I mentioned before, the PhD journey can be very challenging, right? As I say, a lot of failure, but as research it's really rewarding. One is looking back, it's really rewarding, even though it's really tough and challenging. And I enjoyed my PhD journey very much. So there was a saying in Vietnamese saying that failure is a mother of success, right? So basically translated to, you cannot succeed without failing. So don't be afraid of failure. Don't be embarrassed by the failure, right? What more important is you, by the end of the day, you learn about your mistake, you learn about your failure, you pick yourself up, you try again and do it again until you succeed. And that's what I did, right? So bring the title of the talk back here. If I could do it, any one of you can do it. So just go out there and just try, right? And believe in yourself, you can do it. So with that, I want to acknowledge some folks. First of all, at University of Houston downtown, especially Scholars Academy, so for many things, right? As I say, I joined U of H downtown, had no idea that my life would be like this. I will make it this far. So a lot of thanks to Scholars Academy, including all the professors and friends that I made along the way. Another shout out to Dr. Parker. I received the email from her last year, invite you to give a talk, and I was like, all right, this is my dream now. So a lot of friends, when they found out that I was giving a talk at the SRC, they was really jealous, and I can understand why, right? It's a privilege to be here and talk about it. And thank you, Dr. Parker, for thinking of me and invited me to be the speaker. Another shout out for Dr. Jang and Dr. Joes, my mentors during my undergraduate research. Without you guys, I'm not going to be here today. And another shout out to Jerry Johnson. He's my academic advisor. I took biochemistry with him. And I talked to the guy about, grad school, and he like really down to earth guy. He was pretty honest about graduate school, right? And I remember one of the classes during my class with Johnson, I asked him what do I need to do to call him by his first name, and he said that you need to get your PhD to call him Jerry. So I remember clearly the day after I passed my defense, I emailed him right away, say, so I got my PhD now, so I can call you Jerry, right? And yeah, and he's really happy for me. So yeah, thank you, Jerry. We need to catch up soon. So also I want to thank Texas A&M University, especially the Glasner Lab, my PI during my PhD lab. Margaret Glasner, she taught me a lot about research, about enzyme evolutions, and I'm forever grateful. And then Dennis, he was the lab manager during the time, and I learned a lot from him too. And I want to thank my dissertation committee members, Dr. Saccatini, Rochelle, and Dr. Strait, and also thank the Biochemistry Graduate Association, who provide tremendous support for the graduate students to go through their journey. So I'm very grateful for that. And also want to thank Bondwell Technologies and Solugen. And with that, I want to thank you for your attention and listen to my talk today. And I would take any question if you guys have any.
Okay, let's give Dr. Trong a huge thank you. Great talk. Wonderful to hear that. I didn't know that was a saying in Vietnam, or that failure is the mother of success. I really like that saying, right? Because so many of us fail. Wonderful talk. Now, I'm going to take about 2 minutes. We have Dr. Lauren Blanchard here, who will be speaking in just a few minutes. But do we have any questions for Dr. Trong? And Dat, if you will unshare.
Let me stop.
We appreciate that so much. Okay, does anybody just speak out if you have a question? Anybody?
Well, Dr. Parker, if nobody, as an introducer, I do have one question for you, Dad. And then, of course, I know you very well. And then you just gave this one. But it looks like you have been very fortunate to have different mentors and experience or past or advisors. Did you encounter any difficulty, especially when you study at A&M? I mean, this difficulty could be personal, could it be like a research project, or some research project was dead and then you need to actually switch, and then probably wait a lot of time pass. I think a student can benefit a lot from this. And as you said, the failure is the mother of success. And then, yeah, can you elaborate those if you did have?
Yeah, so as I mentioned earlier in my talk, I came here from Vietnam, right? All my parents are still in Vietnam. During my PhD journey, a lot of my friends in my year, they have their family here, right? So they can turn to them, talk to them, just fly home and be with them whenever they have some difficult time, or challenge during, the PhD journey. For me, I couldn't have that luxury, right? My parents are in Vietnam, so all I could do was talk on the phone. But again, it was not the same, right? But yeah, so that was pretty challenging at some points. But you pick yourself up and you get through it, right? But for research, I would say that, definitely there's a lot of failure. I spent maybe 8 months on this single project in the Glasner Lab when I first joined, and it couldn't work. Like, I spent so many time, I tried so many things and it didn't work. And at some point you had to let go, right? Because you could not just like keep doing it and it didn't work. And yeah, that's the one other thing that I learned from grad school, that, you know, you need to learn how to let go. But you learn about the problem, and you know, even though your experiment failed, it doesn't mean that your career failed, right? More important thing is you learn from those failure and you move on with other projects. So yeah,
Wonderful.
I guess it's everywhere with research, right? So
wonderful. Okay, we have a question in the chat. Were you the first in your family to get your doctorate?
actually, I was the first in my family to get my bachelor degree. And yes, and also I'm the first for my family to get a doctorate degree too. So yes.
Wonderful model, wonderful role model. Well, I'm gonna, if you put your questions in the chat, we will come back to them. If that's okay, Dad, if you can hang with us a little bit.
Oh yeah, sure.
I want to take a moment and don't want to hold the president anymore because he's got a lot on his plate, as any president would. We are so happy to welcome Dr. Lauren Blanchard. He's going to share a few words, and I'm going to put you on the speaker view, Dr. Blanchard.
Well, good morning. I can't thank you enough for inviting me here. Frankly, Dr. Trong, I can say to you that everything that I had prepared to say, you embodied and delivered just beautifully in your presentation. I mean, you really do represent that which we aspire to gain more traction here at the University of Houston downtown, and that is, just as you have indicated, understanding the value of undergraduate research. Now, I know that this is STEM research, and I can't begin to tell you, even with the foundation board that I visited with last night, that they are really anxious to make sure that campuses like the University of Houston downtown, with so many minoritized students and many of them who come from underserved backgrounds, that this is the kind of work that they want to make investments in to ensure that we're able to scale you, Dr. Trong, that it's not a one-off, but that it's an overwhelming amount of individuals who then not only benefit and understand not just the value of undergraduate research, but all that comes with it, right? You talked about the mentorship, you talked about the guidance, you talked about the support that you got. And it's like a prescription. And that's why I really appreciated your comments this morning, Dr. Tron, because what you did is that you helped the students to really see from beginning to end. It's because many times students don't see the full picture. And they hear about graduate school, they hear about there is a linkage between the work that they might be doing with faculty in labs and how that could really help you to move into graduate and or professional school. But to go step by step the way that you did today, I just can't thank you enough. I have to tell you that one thing that I chuckled at was the fact that you said that your oral defense, not oral defense, your oral exams, that you found it to be fun. I can say that when I did my oral exams, it wasn't too fun for me. I was kind of overwhelmed with anxiety more than anything else. But bottom line is that I know how important the educational experience is here at the University of Houston downtown. And we take very seriously, and I'm saying this to the students, our responsibility to make sure that we're preparing you with the knowledge, with the skills, with the dispositions, with the experiences that will afford you to reach your respective career goals. And that, when you hear the story of Dr. Trong, you really understand that it's all reachable. It's all doable. Yes, are you going to have some kinks along the way? Are you going to get some no's along the way? Absolutely, you will. But that the interesting thing is that with that strong foundation that we're helping you to build through your educational experience here at the University of Houston downtown, that those no's only become learning moments. And we know quickly that those will be overturned into yeses. So, I wish I could stay the whole day. I mean, what can I say? It feels like my sweet spot. It's my comfort spot to hear not only the stories like yours, Dr. Trong, but in understanding the connection points that you faculty are making with our students to make sure that they really do become the next leaders in their professions. And for me, that's what it's all about. Now, I have to say, Dr. Trong, I was listening very carefully at the end when you made that employment pitch. I just want to make sure that for all the faculty members, that was not for you. We need you here at the University of Houston downtown. That's for our future graduates of our students. And I say that jokingly, but the reality is that, I'm very proud of the work of our faculty, and we need every single one of them in order to make sure that we are producing more and more Dr. Trongs. Thank you for all that you do.
Thank you, Dr. Blanchard, for giving your time and for your kind words. We appreciate it. Okay. Now, I want to go back and see if we had any other questions for Dr. Trong. Anybody? Well, we'll close and just say we are so proud of you because of the success you have made of yourself. And even though you had this supportive system, it really is your own hard work, resilience, and perseverance. And we know there are more students just like you. We just need to plant the seed, and they need to, we would need to water, and then they need to grow. And thank you, Doc, so much. All right, with this, I'm going to turn it over to Dr. Mian Jang, and we will start our oral presenters for the 21st SRC.
Okay, so Dr. Thank you for arranging the whole thing. So I will work with Dr. Parker and trying to just the timekeeper for the oral presenters. Okay, for each of these our 9 oral presenters, you have 8 minutes for presentation, and then followed by 2 minutes Q&A. So let's start with the first presenter. Okay, this will be from Huda Akshikin Oma, and her the title for oral presentation is the role of the transphobia UNC-13 in alcohol-induced hyperactivity and sensitivities. And she worked with Dr. Yuan Kang, and this is her PhD advisors. So Huda, it's your floor.
Thank you, Dr. Zhang, and thank you everyone for this opportunity. If you can just give me 1 second to share my screen.
Absolutely.
Okay, so good morning, everyone. My name is Huda, and this semester I did undergraduate research with Dr. Kang. And for our project, we looked at the role of Drosophila UNC-13 in alcohol-induced hyperactivity and sensitivity. Alcoholism is one of the most important psychological problems today. Ethanol has a big effect on the presynaptic activity in neurons, but it is still not clear how ethanol changes the brain activity or how that change reflects on behavior. One presynaptic protein that binds ethanol is UNC-13. UNC-13 works in the snare complex, which functions to facilitate the synaptic vesicle fusion. And that is necessary in order for the neurons to communicate. One earlier study suggested that when ethanol is present at high concentrations, it binds the UNC-13 and inhibits the binding of its ligand, and that in return affects the synaptic transmission. And this physiology is conserved in both humans and Drosophila. So in order to understand how ethanol changes the brain activity and how that reflects on behavior, we use Drosophila melanogaster, which is one of the genetic model systems used to study alcoholism. Some alcohol-induced responses that we focus on the study are alcohol-induced hyperactivity and sedation. When alcohol is first introduced to the body, we have startled hyperactivity, which is the sudden shock due to the vapor. And then after that, we have the alcohol-induced hyperactivity in this part of the graph. And then after a while, when the alcohol has accumulated in the body, we have sedation. And if you think about it, these alcohol-induced responses are also found in humans. When you first take a drink, like when you first take one drink, you feel hyper. And then after a while, when you consume more drinks, you start getting sleepy, which here we refer to it as sedation. Previous studies suggested that the homozygous for the DUNC13 fail to display neuron transmission and they do not survive. So reducing the activity of the DUNC13 results in flies that are more resistant to the sedation. So we use the heterozygotes, loss of function, mutation, and that reduced the ethanol sedation while also showing a conservation of function between the wild type controls and the DUNC13. When reducing the activity of the DUNC13, we have flies that self-administer ethanol at higher levels in the wild type controls, and that produces a behavioral and physiological resistance to the sedation effect of ethanol. So for our project, our research was to test if the DUNC13 is a facilitated vesicle release, a shared mechanism for hyperactivity and sedation. To test our research question, we used two assays. The top one here is called the fly bar loss of writing assay, which was used to examine the sedation effect of ethanol on DUNC13 compared to the control flies. In this assay, the flies were exposed to ethanol, which passively enters through their respiratory system and increase their internal alcohol concentration. And that would result in more flies that lose their writing reflex with time. For this assay, we used 30 flies in each of these vials, and the water and ethanol concentrations were controlled using the flow meter and the air pump. And for this assay, we counted the sedated flies using 5-minute intervals, and the fly was counted as being sedated if it had fallen onto its back or side and could not right itself. And for this assay, we used the t-test for statistical analysis. And for this second assay, it's called the FlyGram assay, which stands for Fly Group Activity Monitor Assay. This here is the software that was used to measure the hyperactivity of the flies. And these are the arenas where the flies were loaded. And these were graphs that were generated by the software and represent the number of active flies versus time. In this assay, we loaded 10 flies in each of these arenas, and we set the program was set for a total of 20 minutes. For both of these assays, we used DUNC13 and control flies, both males and females, and the flies were between 2 to 5 days old. From the fly bar loss of writing assay, we found that the heterozygotes for DUNC13 were less sensitive to the ethanol sedation effect. In this graph here, the T-half is defined as the time half of the flies lose their writing reflex, which means the higher the T-half, that means the flies are less sensitive. That means it takes longer to get sedated. We found that the DUNC13 males and females took longer to get sedated compared to the control males and females. From the FlyGram assay, we found that the DUNC13 flies have higher mean of alcohol-induced hyperactivity. The graphs here represents the average of active flies versus time in seconds. And this part of the graph, the flies were only exposed to water. And this bar here is the time interval where the flies were exposed to ethanol. Yes, and the results were consistent for both males and females, and no statistical analysis has been done yet. So when doing the experiment, we hypothesized that the DUNC13 mutant flies were less responsive to the alcohol-induced responses. We found that the DUNC13 mutant flies had more resistance to the alcohol sedation in both males and females, and that was consistent with the DUG13's role in synaptic release. From the flygram assay, we showed that the data showed that the DUG13 mutant flies may have had a higher response to alcohol-induced hyperactivity, suggesting that it may be a separate mechanism. So when doing the experiment, there was strong evidence that there was specific changes in the presynaptic activity underlying alcohol-induced responses. Some factors that we believe affected the experimental outcomes are ethanol concentration and unequal sample sizes. So we need to validate our data by repeating the experiment with a higher sample size and with optimization of the ethanol concentration. We are also working on a different assay that measures the speed of the flies, and that would also investigate the role of DUG13 in synaptic vesicle release. This study is very important because it is one way to understand the role of DUG13 in the presynaptic activity. And once we fully understand the role of DUG13 in alcoholism, we can use these kind of studies to produce medicines that could target DUG13 and cure alcoholism. Before I end my presentation, I want to acknowledge Dr. Greg Rahman. He's a professor at the University of Mississippi, and he provided the flies for us. And Dr. Kang, she was a great mentor, and I have learned a lot from her. And of course, Scholars Academy and Dr. Mary Jo Parker, the executive director. We also want to thank the MSEIP grant and the other lab members, Joel, Evelyn, Michelle, and Thomas. And we also want to thank the previous UHG students who also worked on this project, and they contributed to this outcome, Jose Gonzalez and Daniel Adrian. Thank you so much for listening, and we can go ahead and proceed with the Q&A portion.
Thank you, Huda. Yeah, any questions? It's a wonderful job. Yeah, it's so productive and just one semester's work. Yeah.
Thank you.
Okay, can you unshare, Huda? Wonderful, wonderful work. Okay, do we have any questions? Anybody, students? It's 10, you should be awake now.
I have a question.
Okay, great. Hatun?
So you mentioned that you think it might be a separate mechanism. I want to ask why you think that might be and what that mechanism might be.
Yes, it's because we use the same heterozygote flies for both of the assays, the sedation and for the hyperactivity. So, so we would expect that for both assays, we would see less response, but in the hyperactivity, we saw a higher response. So we might think that there might be like other interactions, like maybe the DUG13 was interacting with other proteins, hormones, or maybe that response was in different types of neurons. So yes.
Okay, we have one more question. I'm going to paraphrase it. So Janiyah Joseph would like to know, is the effects that you found in flies, would they be relatable in some manner to humans in terms of alcohol? Can you speak to that?
Yes, yes, they are. You can see these kind of responses also in humans because, like I said in my presentation, when you first take one drink, you start getting hyper, like you want to go take a dance, you know. Yes, and after a while, when you take more drinks, you start getting sleepy, and that here we refer to it as sedation.
Okay, excellent. One last question. Let's see, can you visualize to determine the flies that were DUG13 heterozygotes, or is it you buy them that way? Can you speak to that?
Oh, yes. Actually, it was a really long procedure. We actually did crosses. So we had the DUG13 mutant flies and we cross it with the wild type. And then we have the genetic marker, and then we can select for the heterozygotes because these are the flies that we want.
Excellent. All right, everybody, let's give her a hand. And if you want to know more about that, she'll be presenting a poster during the poster session. Thank you so much. All right, let me introduce Our next presenter, Ms. Hatun Badwadi. Her title is Variability of Traits Conferring Drought Tolerance in Prairie Grasses Along a Precipitation Gradient. Co-authors are Alana Barrera, Huda Omar, and Arnold Villatoro. Her research mentor is Dr. Michael Tobin. And Hatun, we're going to give you the presenter view, speaker view, and if you'll share your screen.
All right, I'll do that right now.
Thank you.
All right, can everybody see my screen?
Yes.
Yes,
Great.
good.
Okay, so thank you for introducing me and for letting me present the research on the ongoing study of variability of functional traits in prairie grasses. Before we begin, I'd like to acknowledge the help and support of my UHD undergraduate collaborators and those who were able to fund this project. And I'd also like to thank Scholars Academy for their support as well. To start with, it's important to understand what functional traits are. They're clearly defined characteristics of plants that strongly influence their growth, reproduction, or survival. One example is seed mass. It can interact with ecological factors, biotic and abiotic, that determine its dispersal and ability to germinate. Many studies focus on the variability of functional traits among different species, but functional traits can also exhibit variability within individuals of the same species. If you look at this diagram on the right over here, we have environmental gradient on the x-axis and functional gradient on the y-axis. And at different levels of an environmental gradient, the performance of the functional trait can change accordingly. So at the peak is optimum performance. And it's important to understand functional traits, and that's because of how they can be used to further understand and model ecological properties, which are the structures and processes of an ecosystem. By studying these functional traits, we can understand the plant's tolerance to environmental stressors and how they differ across a resource gradient. We're interested in drought tolerance, so we focused on water loss from the leaf. And a major route for water loss is through the stomata, which are small pores in the leaf surface that allow uptake of CO2 from the air. Water evaporating from surfaces inside the leaf diffuses out of the leaf via these stomata, and they diffuse out as water vapor. This is a process that's called transpiration. Plants need to take in CO2 for photosynthesis, which is a process that creates oxygen and energy. And as you can see on the diagram here, while the plant is taking in CO2, H2O is being released. So there's a trade-off between taking in more CO2 for photosynthesis and preserving water in the leaf. And that's also indicated by how the arrows are going in opposite directions. In this specific project, we focused on stomata, which are found in the epidermis of the leaf. And in addition to being responsible for gas exchange between the plant and the environment, they can be opened and closed by guard cells that border the stomata. When these are closed, there is no CO2 being taken in for photosynthesis, but water loss is greatly reduced. And so the features that we measured were stomatal density and guard cell length. Not only did we measure stomatal density, but we did it across the precipitation gradient of Texas. If you look at this map here, you can see that it rains more in the east than it does in the west. And the arrow shows the direction in which we took samples. The species that we chose for this project is Schizachyrium scoparium, also known as little blue stem. It's a dominant component of the tallgrass prairie native to eastern Texas. Plus, it's also found along the precipitation gradient, making it practical for sampling. To bring it all together, we investigated the variability of stomatal density and length across the east to west precipitation gradient in Texas. Samples were collected during 2 time periods, which are the spring and late summer of 2021 at 5 to 6 sites along the gradient. The map over here shows the locations of the sites that we sampled from. Impressions were made by painting a very thin layer of clear nail polish and allowing it to dry. And then using clear tape, we lifted that nail polish impression off the leaf and taped it to a slide for observation. The image on the left shows what stomata look like at 400x magnification, and they're pretty evenly distributed throughout the whole image, but they're especially easy to see closer to the bottom. And I have one circled in red for you all. You can see the guard cells that border the stoma as well. Before we started counting stomata, we made sure to check the impressions from the upper surfaces of the leaf, because in little blue stems, stomata should only be on the lower surface of the leaf. And this was just an extra precaution to ensure that we had the correct species. We counted stomata by choosing 2 random areas on either side of the midvein, which is a central vein that runs down the leaf. The image on the right shows a visual of what that looked like. And so that made a total of 4 fields of view per sample. Then we took images of the stomata, which we used to measure the lengths of the guards. Any data that we had was recorded and put into a spreadsheet and evaluated. So now on to the results of the data that we worked with. Our first graph over here is stomatal density for the spring sample. On the x-axis, we have precipitation in millimeters, and on the y-axis, we have stomatal density in number per millimeter squared. There is a relationship between the 2 variables. As precipitation increases, the stomatal density decreases. The next graph here is a stomatal density for the late summer sample. Once again, we have precipitation on the x-axis and stomatal density on the y-axis. There was no relationship between these 2 variables. And for our final graph, we have stomatal length for the late summer result. And we have precipitation on the x-axis, and we have stomatal length in microns on the y-axis. There is a relationship between the 2 variables. However, it was just marginally insignificant. It is evident that there is a variability of functional traits among individuals of the same species, which was related to precipitation in some cases. Since this is an ongoing study, we will also analyze the widths of stomata and minimal leaf conductance in order to further understand drought tolerance for this species. Thank you all for your attention, and I'll be happy to take any questions now.
Okay, thank you very much, Hatun. Let's see, can you unshare? Great. Now, do we have any questions? Anyone?
I have a question.
All right.
Hatun, why do you think there was no relationship between some of the variables in the late summer results?
Okay, so as you can see, there was only only the spring sample showed some significant relation between the 2 variables. And for the summer, we're taking into consideration the fact that it might be something beyond the plant adapting morphologically according to how it can tolerate drought. So it might be an avoidance strategy that the plant has developed instead. So when there is drought, it just won't grow. And if there is sufficient water, it will grow, but there won't be variation according to the gradient. So that's something that we're looking to as well.
Great. Anyone else? Any other question?
I have a question.
Okay. Sean.
So my question is, was there any sort of measure of data analysis of like the stomata density from site to site? And if not, do you have any plans to do so?
So the stomatal density wasn't just for one sample, it was for several samples, and we had a plant from every site. So this relationship that we saw between the precipitation and the stomatal density was going across a precipitation gradient. So from those 5 sites that I was talking about earlier.
Okay, great. And I want to encourage everyone, we have the chat open, and you can direct any other additional questions you might have for our keynote speaker. You can direct to him. I'm sure he's monitoring that, and to all of our oral presenters as well. For the sake of time management, we're going to kind of limit to 2, maybe 3, and we'll move on. All right, Dr. Jang, if you'll introduce our next.
Okay, yeah, so wonderful job, Captain. As Dr. Parker said, you can continue on the questions in the chat room. So yeah, I'm glad to introduce our third speaker, actually speakers, no, the 2 presenters, Anna Sherbarsky and Annie Wu. And then their titles for the research is testing degradation of biodegradable plastics. And the research mentors Dr. Polan Gulati Sahotra. So Anna and Annie, so now it's your platform now.
Okay, so I will share my screen now. Okay, can everyone see my screen?
Yes.
Yes.
Okay, great. Okay, so hello, my name is Annie Vu.
Hi, my name is Anna Sobolski.
And today we're going to present our research on testing the degradation of biodegradable plastics. So first off, learning about how plastics degrade is a very important topic because of the growing pollution problem around the world. There are more than 380 million tons of synthetic plastics that are produced annually worldwide, with a decomposition rate of up to 500 years. Although plastics can be reused or recycled, they do not have a good retrieval rate, which means that recycling and reusing are not our permanent solutions. As opposed to these physical and chemical processes, the best way for plastics to break down is by the natural processes of bacteria and microbes. And research has shown that microbes are able to attach and form biofilms on moist surfaces of plastic waste. And biofilms form when microbes are able to attach to each other and are often able to attach to various different surfaces as well. In this research, we are using biodegradable plastics as opposed to your more common synthetic plastics. And this is because biodegradable plastics are made to be compostable with the aid of microbes. The goals of our research are to identify which soil microbes are involved with the degradation of the biodegradable plastics. We also determine any changes over time in the soils, as past observation has shown that microbes in the soil have plastic degrading abilities. For our materials and methods, we use 3 different types of biodegradable plastics. They are all known as polylactic acid plastics, or PLA for short. If you look at figure 1 on the bottom left-hand corner, they show the plastics that were prepared in lab, and this is how the plastics looked before receiving any soil treatment. So we test these plastics in 4 different types of soils as shown on figure 2. They are Miracle-Gro potting mix, Miracle-Gro performance organics, Timberline cow manure and compost, and farm dirt. So samples are left in the potted months anywhere from 1 to 6 months. When the samples are collected, they are cleaned and scraped for microbial biofilms. These are then frozen as pellets and sent for identification using DNA sequencing. We also observe and record any physical changes that are seen in the plastics and use infrared spectroscopy to identify any chemical changes in the plastics as well. So results in discussion, physical changes. After pulling plastics from the soils, there are changes that can be seen that are equivalent to degradation taking place within the soil. So the plastics look duller than before they were placed, and some plastics also show signs of curling, cracking, and any texture changes. So if you look at figure 4 and figure 5, these show our control samples for plastic type 2 and type 3. And then if you look at figure 6, this is our farm dirt sample 2 that was collected on February of this year. Compared to its control on figure 4, you can notice that the plastic is duller. Figure 7 shows farm dirt sample 3 that was also collected from the same date as well. Compared to figure 5 of the control, this sample shows noticeable curling around the edges.
IR spectroscopy. Plastics from 2 of the 4 soils tested show noticeable changes via IR. The 2 soils are timberline cow manure and farm dirt compost. Results come in the form of hydrolysis taking place on the plastic sample as shown in the first circle on the top left. The bottom circle shows the carboxyl group decreasing. Figure 7 is the sample 3 plastic control IR, while figure 8 is the sample 3 farm dirt IR. The top left circle illustrates the hydroxyl group on the IR spectra. Figure 7 shows a straight line, while figure 8 shows a prominent dip. The bottom right circle shows the carboxyl group on figure 7. The sort is very long, while on figure 8 it shortens quite a bit. The region on the right after our carboxyl group is the fingerprint region, which is not usable data in the case of our experiment. All plastics had physical changes, including shape, distortion, and color. The soils had more change in the time allotted, which were farm dirt compost soil and the timberline soil because of higher microbial content. The Miracle-Gro soils showed considerably less progress during time in the soil. Sample 3 in the farm dirt compost was shown to be the one with the most changes having taken place. This sample is a polylactic acid plastic made from corn grown in Nebraska. Future steps. Future steps include placing samples in the garden beds at the Science and Technology Building at UHD. Previous research has shown that different parts of the plant, such as the leaf surface, have different microbes that may assist in the degradation process. In addition, plastics would have exposure to plant roots that also possess microbes which could help in degrading plastics. Exposure to these different pieces of the plant would give the plastic more opportunity for degradation. Previous research has shown that fungi presence on leaves is a possible degradation assistant as well as microbes within the soil. We wanted to say thank you to Dr. Galati for mentoring us as future researchers in science. Thank you to Scholars Academy for allowing us to present our research. Thank you to Dr. Joes for advice regarding our research. Thank you to the Department of Natural Sciences for providing great lab space. Thank you to our lab mates, Sabrina and Eunice, for being a supportive other half of the lab. This work was supported by the Welch Grant and the Orca Grant.
Okay,
Question?
Yeah, thank you, Annie and Anna. Wonderful job. Any questions?
Yes, I have a question.
Please.
Okay.
Have you guys identified what microbes in the soils that contribute to the degradability of the plastics?
We have not been able to identify microbes yet. We've only been able to identify which chemical processes are beginning to take place.
Sounds good. Thank you.
I wanted to ask, the farm dirt, I believe you said, had the greatest results.
Yes.
What was the timeline for getting those results?
So our plastics were left in the soil anywhere from 3 to 6 months. The greatest results were left in the soil anywhere from 7 to 8 months. It was the first time during the experiment process we had left them in there that long.
Okay, wonderful. Thank you so much, ladies.
Thank you.
Thank you.
Okay. Move on to our next presenter, Ms. Kai Lu. And yes, you're next. I'm introducing you. Her title is Using Therapeutic Chinese Massage to Treat Peripheral Neuropathy in People with Human Immunodeficiency Virus, a randomized controlled trial pilot study. Her co-authors are Dr. Linda Doon, Min Zhang, Lucy Debussy, Carlotta Sweeney, and Shi Wei. And her research mentor is Dr. Song Ji. Okay, so we're going to put you on the speaker view, and if you'll share your screen.
Okay, thank you. Let me share the screen.
Okay. Wonderful.
Can you see the screen?
Yes.
Okay. Thank you. Good morning, everyone. Thank you so much for your attention to my presentation. This is Cai Liu, a student in the USD RN2BSN nursing program. My presentation today is on using therapeutic Chinese massage to treat peripheral neuropathy in people with HIV, a randomized control trial pilot study. Dr. G is our mentor in this project. My presentation will include 6 parts: the background, problem, purpose, methods, results, conclusions, and the implications. Let's start with the background. In the US, more than 1 million people are living with HIV. Peripheral neuropathy is one of the most common neurological disorders in people with HIV. It occurs in half of the people living with HIV. It is characterized by burning, stiffness, prickling, tingling, and numbness, or a loss of feelings in a person's extremities. Currently, there is no FDA-approved treatment for HIV-related peripheral neuropathy. This is the problem. People with HIV and peripheral neuropathy have to take anti-seizure or pain medications to temporarily relieve their symptoms. Only a small portion of people who take medication achieve a reduction in peripheral neuropathy symptoms. Moreover, research in this area has been very limited and has not progressed well. Chinese massage has been proved to offer many health benefits, such as improved nasal breathing in children and reduce lower back pain in adults in previous research studies. Massage has a lower relativity risk if delivered by qualified personnel. Thus, it can be a low-cost, low-harm, and effective approach to treating HIV-related peripheral neuropathy. In this study, we propose a randomized control trial pilot study to examine if a series of 3 weekly 25 minutes therapeutic Chinese massage by specialist provider could reduce peripheral neuropathy-related pain and improve lower extremity functioning in people with HIV. The study is funded by the UHD OCR grant. It gained support from the AIDS Foundation Houston. Participants were recruited by referral of the staffs, as well as snowball method. The research team has 2 sites to the HIV Foundation Houston between December 2021 and January 2022. Our participants must meet the 6 inclusion criteria to join the study. People with confirmed HIV and peripheral neuropathy symptoms on their lower extremity. People not currently take medications to control peripheral neuropathy symptoms. 18 years and older, English speaking, can have rational conversation and not pregnant or lactating. We randomizely assigned the participants into one of 2 groups, the treatment group or the control group. The treatment group received 3 weekly 25 minutes Chinese massage on their lower extremities. The control group received 3 weekly 25 minutes placebo massage provided by the same provider. The participants did not know which group they were in. They were blinded. Dr. Dunn provided the massage as well as the placebo massage. As she is a specialist in Chinese massage, the massage included acupressure to the points, along with the distal lower extremity acupuncture points of the gallbladder channel. Further Chinese massage includes techniques of needling, rolling, rotating, pulling, and scrubbing to the lower extremity. The control group included gentle rubbing to the foot and the toes without point stimulation or other techniques of Chinese massage. The control group received verbal instruction about how to perform the massage after the study was completed. We assess neuropathy pain and lower extremity function prior to and after the intervention for both groups. Neuropathy-related pain was measured by the numerical pain scale. 0 means no pain, 10 means the worst pain. Lower extremity functioning was measured by the lower extremity functional scale. The higher the score means the better functioning. We use the iPad to collect the data. From the table, we can see that most of the participants were middle-aged African Americans. All of them lived alone. They have lower socioeconomic status. For statistical result, paired t-test showed that pain level was reduced and the functioning of lower extremities was improved in the experimental group after they received the intervention. Although the result was not statistically significant, it may be due to the small sample size. We are continually recruiting more participants for the study. The implications are that Chinese massage may improve lower extremity functioning and reduce pain among people with HIV. More studies, including another sample size, are needed to examine the relationship. Thanks again. Please, if you have a question, please ask me. Thank you.
Go ahead. Do you want to unshare your screen, please?
Okay.
Very nice. Do we have any, do we have any questions? Well, I do have one for you. You know, the self-report, did you consider, well, you know, there's biases with that, but did you consider any type of invasive measurement, maybe of blood or white blood cells, you know, that tend to increase with stress levels or other, just curious.
Because pain is very subjective. Pain is subjective. So it sometimes it is difficult to test by blood or other thing because it is very subjective. Different people maybe have different feeling.
Okay, just to follow up very quickly, did you think about an EEG to measure as a way of measuring possible impact of the pain or levels of the pain? Just curious.
Oh, yeah, it is a good idea. If pain, maybe his heart rate maybe increase and the EEG maybe have some difference. It is good idea. Thank you.
That's a great, really wonderful study and very necessary. Do we have any other questions from anyone? Let me see the chat real quick. I did not see, I was asking my own.
I do have a question for
Okay.
Kai-Ying. It's a very interesting talk, and it's quite different from actually the rest of the presentations. So that's why I make it unique. I personally view this treatment or the studying. So it's kind of like a pain relief or neuro-related studying, right? So there are several ways, and then a completely just a spiritual or long material-based one will be like a meditation. And then another one will be, as Dr. Parker said, it's like a invasive, like a blood drawing. So that's kind of very aggressive treatment. And then your presentation here, Kai-Ying, is like in the middle. It's like a more massage, it's more external, it's not going to take a pill or medicine. That's quite interesting. I think that's an alternative way to treat the whole thing. Then my question for you is, and for Dr. Ge is, by the way, her name is Dr. Ge, not Dr. G.
Oh, I'm sorry. Apologies.
Yeah, fine. Yeah, a lot of, so take this a chance to try to correct that. And then, so did you, in your study, did you ever consider to compare these three kinds of things, like the very aggressive and non-aggressive at all, like a meditation, and then your way, then trying to find a relationship between these three? I mean, of course, they may have both pros and cons. Did you ever consider about those?
We will consider it, and this is just the beginning for the research. Thank you for your suggestion. We will consider it.
Good. Okay.
Okay, very nice. Thank you so much.
Okay, any more questions?
Any more?
Yeah, if no more, then as Dr. Parker already suggested.
Question.
Okay.
Yes, what is acupuncture and how is it helpful in reducing pain?
Oh, the gallbladder channel, Chinese medicine believe the gallbladder channel runs from your fourth toe, through your foot, up your leg, through your trunk, and to the top of your shoulder. There is one gallbladder pathway on each side of your body, and there are 44 acupuncture points along this channel. And the Chinese medicine believes that it is related with lower extremity pain and numbness. So we chose the gallbladder channel.
Okay, thank you. Okay, Dr. Mian, would you like to introduce our next presenter?
Sure. Okay. So yeah, our next presenter will be Robert Lacroix. His title is Discovering New Compounds for Anti-Cell Proliferation Activities, and his research mentor is Dr. Rachna Sadana. So Robert, now is your floor. Please show your screen.
Okay. Are you able to see it?
Yeah, we are able to see.
Great. Okay, so I want to introduce y'all to my SRC project titled Discovering New Compounds for Anti-Cell Proliferation Activity. And so let's get started. And so some background information on this project. So a common treatment for cancer is the use of chemotherapy drugs, but this isn't really the safest treatment, as can carry risk to the user, as can usually cause side effects. And the most common side effects of chemotherapy drugs include nausea, vomiting, hair loss, and fatigue. And so the goal of our research is to find new chemotherapy drugs that can minimize the amount of side effects that it would inflict upon the user. And so to do this, I mainly utilized two experiments. And so the first experiment is called compound screening. And the purpose of compound screening was that we we got some synthetic compounds, and we wanted to test them on how well they can inhibit the cell growth of different cancer cell lines. And so with that, and how we did that was, so the first I plated the optimal amount of cells, which was around 50,000 cells were loaded into their wells. And then for precision, I duplicated the wells. And then in each well, I added 10 microliters of the synthetic compound in their own respective well, as well as two controls being PBS oxyrubicin. And then I let that incubate for 2 days. And then I added 10 microliters of MTT or added per well, and then let incubate for 3 hours. And then 2 hours later, I added a detergent reagent. So the purpose of the MTT, so as you can see the picture on the bottom, is that when you put it in the wells, it's yellow. And then you let it incubate, then you add the detergent reagent after the incubation, and then it will turn purple when it detects mitochondrial dehydrogenase, which is found in living cells. And so basically, it's a good visual indicator if cells are alive or not, because if the well turns from yellow to like a really dark purple, then you see, oh, there's like a lot of cells that lived, or if it's vice versa, if it's yellow or more yellow, you can see, okay, there's more cells that died. And so, but I also read the plate in the spectrophotometer to get absorbance numbers, which I then use for in the results. And so, as for the second experiment, is the apoptosis necrosis assay. And so, from the compound screening, I used the potential compounds that look potential in inhibiting apoptosis necrosis, and I used it in this assay. And so, basically, the purpose of this assay is to figure out if the cells died via apoptosis or necrosis. And apoptosis is a natural cell death that we want to happen, while necrosis is quite the opposite. It's an unnatural cell death that can cause inflammation, which is what we don't want to happen. And so for this assay, what I did was I plated 100,000 cancer cells into their own respective wells, and then I treated them with the promising synthetic compounds that I learned from the previous experimental compound screening. And I let incubate for 48 hours. Then I added visual markers to then visualize the results with the fluorescence microscope. And so now for the results, these are the results for the compound screening on the MOLT4 cell line. And the MOLT4 cells are leukemia cancer cells. And so in this figure, it shows the effect of the 30 compounds coded the PM series on their proliferation on the MOLT4 cell lines. PBS was used as a negative control, while doxorubicin was used as a positive control. And then each synthetic compound was used at a final concentration of 20 micromolar. And so here, what I'm looking for is mainly for compounds that can inhibit the average cell survival rate into the 50% range or less. And the ones I want to pay close attention to is PM9, 10, and 11, as they as they get the average cell survival rate into the 50% range. And there's other PM series that do this, like 27 and 3, but I've done this experiment multiple times, and PM9, 10, 11, and PM9, 10, and 11 are the most consistent doing this. And so I also did the compound screening assay on the BXPC3 cell line, which is a pancreatic cancer cell line. And you can see the PM series compounds are, you can see they're more effective on this cell line. But then again, I want to pay attention to the PM9, 10, and 11, since again, they're consistent between both cell lines and inhibiting the average cell survival rate into 50% range or less. And again, the same controls are used, PBS as negative control and doxorubicin as positive control. And so, and leads me to my results for my apoptosis necrosis assay. And so in this assay, I chose to test PM9, 10, and 11 to see whether or not they induce apoptosis or not. And so again, so what you're seeing is that so blue dots represent alive and healthy cells, while green dots represent cells that went through apoptosis. And if you were to see red, that would indicate they went through necrosis. And so as you can see, PBS, as it was a negative control, you can see that it's working as well. There's a lot of cells, which is to be expected. And I used camptothecin as a positive control, and it did its job. As you can see, there's a lot of green dots indicating apoptosis. But then you can look at the PM 9, 10, and 11 compounds, and you can see there is apoptosis going on, but it's quite similar to the PBS. And so that kind of leads me to believe that apoptosis is not occurring with these compounds. And so in conclusion, so the PM compounds 19, 11 are promising in inhibiting the proliferation of MOLT4 and BX species cell lines. This is mainly because they're able to get the average cell survival rate into 50% range or less. But as shown in the apoptosis necrosis assay, those compounds do not seem to be inducing apoptosis. In the future, I plan on testing these compounds on different cancer cell lines and testing new compounds and see how they can compare. And acknowledgements, I want to thank Dr. Sadana for being such an amazing research mentor, giving me a chance and like being like super cool. I love you. And then, and also I want to thank Andrea Guzman. She basically taught me all of my like techniques. And I would also like to thank Orca for funding it. And thank you for watching. And yeah, that concludes my presentation.
Thank you, Rob. Any questions?
Okay, let's see. I thought I saw a question. Just a minute. Let's see. Christina Wynn has a question. Would you be testing other cell lines, not lymphoblasts, such as MCF7, breast cancer cells? Was her question.
Yes. We're actually doing that like right now. Yeah, yes, we are doing it on other cell lines like MCF7 and A549, which is a lung cancer cell. And okay. Yeah.
Great. Any other question? Okay, keep working hard. And
Wonderful
we'll move.
job,
Thank you so
Robin.
much for your presentation.
Thank you.
All right.
Thank you.
Now we'll move on to our next presenter. Kayla Mazarigo with a title presentation, The Effects of Recidivism on Residents in Halfway Homes. And her research mentor is Dr. Judith Harris.
Good morning, everybody. Can you see me?
Yes.
Okay. So really quickly, I wanted to really formally introduce myself before I got started with my presentation. So my name is Kayla Mazariego. I am a third-year student at the University of Houston Downtown, majoring in criminal justice and minoring in political science. And I am the Newman Civic Fellow at UHD. This means I am the official student representative for the University of Houston Downtown. Now that I've formally introduced myself, I would like to get started with my presentation. Okay, can we all see it? Okay. So this is halfway houses and recidivism. So what exactly is recidivism? Recidivism is when an individual is released from a correctional facility and reoffends. In this study, I specifically focused on halfway houses. A halfway house is actually a form of transitional housing for individuals coming out of incarceration or seeking help before they are incarcerated. Now, the real question is, what do halfway houses do for residents? Now, if someone suffers with drug or alcohol addictions, they are put into halfway houses that offer rehabilitation programs that help with substance abuse to prevent further chaos within themselves. Why is recidivism such a big issue? Well, when individuals reoffend and are reincarcerated, this keeps the population of imprisoned individuals high. This can lead to jail and prison overcrowding, and Houston is currently dealing with this problem at the moment. Prison and jail overcrowding can be very difficult to handle. What are the main causes of recidivism? Now, the best explanation, according to Tiging and Abadi, is the relatively high recidivism rates are among released offenders were centered on the offender's educational illiteracy, lack of vocational job skills, lack of interpersonal skills, or even criminal history. Many issues that contribute to recidivism are substance abuse, mental health issues, lack of money, housing, delinquent peers. All of these issues can lead to incarceration, depression, drug addictions, not being able to finish school, not having an outlet, not having something, no more passion, motivation for things. My literature review is based on, so there is actually a substantial lack of research on documentation regarding the recidivism of residents in halfway houses. Conversely, there is considerable amount of information regarding the recidivism rates of correctional facilities. The statistics do show a disturbing trend. However, the reality is that there's little to no data on individuals sent to halfway houses after incarceration and whether they contribute to recidivism rates. According to the National Institute of Justice, approximately 44% of criminals released commit an offense within 1 year of release. Now, Delaware currently has the highest recidivism rate in the United States at 64.5%. According to the State Statistical Analysis Center, as of 2020, 65% of people released from the Delaware prison in 2016 were convicted again within 3 years of their release. This is a map of UHD that of the Houston area that I created. So all these blue dots are actual halfway houses in Houston. You can see how close many of them are to UHD themselves that we are not aware are even halfway houses when we walk past them in downtown. According to my preliminary research, there's about a halfway house between 10, 15 miles. And we can really see right there and focus on the southeast part of Houston, how many halfway houses there are. Now, they're scattered everywhere, of course, because if somebody needs help, they need access to something that's close to them. And it's just really important to have an image in mind to know in order to understand why this research or these studies like these should be conducted. Now, this is the Texas Department of Criminal Justice information. This study is a 10-year study that was done from, I believe, 2005 up to 2015, but they recently got the information in 2020. So the ultimate goal of the TDCJ's reentry program is to reduce recidivism completely. Recidivism rates for TDCJ offender population continue to be among the lowest in the country, with currently overall, it says 20.3, but up to date, as of recently this year, it's moved up to 22.6%. It is still among the lowest in all of the United States. But as of June 30th, 2020, approximately 411,629 individuals were on probation, parole, or were incarcerated. 201,120 were on direct misdemeanor or felony probation. 126,590 are incarcerated currently in TDCJ facilities. 83,423 are under parole supervision. Now, throughout Texas, over 1 million of offenders are processed in local jails each year, and many of whom are in transition to TDCJ supervision. Now, these statistics are based on a lot of research from peer-reviewed articles. So Texas has a low reincarceration rate compared to other states. And according to the Texas Commission on Judicial Conduct, within Texas, 21.4% of individuals released from prison actually return within 3 years following their release. Now, TDCJ offers a 3-phase reentry program. And this is phase 1, identification processing, which is, it assists eligible offenders in obtaining replacement social security cards, certified birth certificates, state identification cards at the time of the release. Phase 2 is assessment and reentry planning. This is a completion of risk and needs assessment with an individual case planning provided to those at moderate to high risk of reoffending. And phase 3 is community reentry services. So here it provides post-release individual cases, training, and employment services with an emphasis on assisting the formerly incarcerated with obtaining substantial employment. This is very important because after individuals are released from incarceration, they have nowhere to turn to Many of them cannot get a job because of that label that is being put on them upon release. They serve their time and now they can't get a job. And the reason why it's an issue is many turn back to the life of crime that led them to incarceration in the first place. So reentry programs such as these, the programs that TDCJ offers, help individuals in finding a stronger ground in order to hold on with what life throws at them. Now, nationwide recidivism rates have remained high for the last decade. Roughly 67% of offenders released from state prisons return to the system within 3 years. This is information acquired from the US Bureau of Justice. Now, since the start of the pandemic, getting recent information and data has been increasingly difficult. It's been difficult to figure out how many people are going back into prison after being incarcerated. It's difficult to get that information because COVID made it difficult. We couldn't track individuals like how we used to. But 3 out of every 4 released inmates will be arrested again after release, and over a half of all inmates will return to prison soon after release. Now, I don't know about many of you, but those numbers are scary. Now, 49.7% of offenders return to prison within 3 years after release, and 55.1% return within 5 years. So halfway houses help individuals, like I said, by giving them a solid ground to stand on, but also someone to talk to, therapy, offering programs in order for them to not feel alone. Many individuals who are released from prison have lost their families or haven't seen their families because they were pushed away from them because of the decisions that they created. So many halfway homes, private or government, they make sure to help these people feel whole again, because that is what they deserve. And in conclusion, it is difficult to discern and pinpoint an exact cause of recidivism. And until further research is done and a visible link can be seen between halfway houses and residents, we cannot see truly if residents contribute to the ongoing growing number of recidivism. Now, Houston, although it does have among the lowest in the country, Delaware the highest, we need to be asking ourselves what can be done to reduce recidivism completely as a community. And without that, then these are my references. And thank you so much.
Okay, thank you. And if you'll unshare.
Yes, ma'am. Stop share. There we go.
There you go.
There we go. Thank you.
All right. Do we have any questions? Nice job, Kayla. Any questions, anyone? Well, I have one, if I might. So did you find any information regarding the neighborhoods where they're putting the halfway homes?
Now,
where they don't necessarily want the halfway house to be located there. Is that, how much of an issue is that in this particular problem? Or did you investigate that?
No, I actually did. So many, if we were to visualize the map right now, many of them are scattered in areas that you would think, why is there a halfway house there? It's kind of a, it's kind of an expensive area, but there have been a lot of issues where neighborhoods do not feel safe knowing that there are criminals down the street, even though they serve their time, even though that they're done with that. And many people fail to realize the extent of how important it is to help others and make sure that us as a community can stay enforced because we don't know what others are going through. And maybe, you know, me personally, if I were in that situation, I live next to a halfway house that I didn't even know was a halfway house at the time. And that was shocking to me. And it really opened my eyes to how blind we can be sometimes just because of this label that somebody receives.
Right.
So,
Okay, I'm
yeah.
gonna call you there, and thank you. Let me read, I'm gonna give Dr. Jang the last question, but let me read one from Omar. And his question was, or maybe it's praise. No, let me see. Had a couple here.
I believe it's Omar.
Okay, can you see it?
Yeah, it's he asked,
Go ahead and read it.
Sobriety a requirement for these halfway houses, and are they linked to recidivism and drug crimes? That is actually, I'm really glad that you actually asked that, because sobriety is a huge thing in order to be eligible to remain in a halfway house. Now, in a halfway house, they give you many rules. It's, you know, my house, my rules. It reminds me of my mom and me living there. So if you want to remain in the halfway house, you do have to remain sober. You can't bring drugs in, you can't bring alcohol in, you can't bring things that are going to influence other residents, because if not, you are at risk of getting kicked out and being returned and reincarcerated once again, because either you break parole, or possibly they're on probation. It all depends on the situation, and it can be very circumstantial, but many of these halfway houses offer do have those rules where you have to remain sober. They have curfews and all of those things. I also do believe that there are some situations where, so there's silver living and then there's outliving. So the silver living individuals are to stay inside the halfway house at all times. They are not permitted to leave. They live inside the halfway home. Now,
okay, and I'm gonna stop you there. Great answer. I want you to go to the chat and you can answer. There's a couple of other questions, and you can expand this answer for Omar, but also address the other question. Very nice presentation. Very nice.
Thank you.
All right. Dr. Jang, if you want to introduce our next presenter.
Okay, yeah, very interesting presentation, Kayla. The halfway house, I think it's a very eye-opening for that. Okay.
Thank you.
Our next presenter will be Bianca Mendoza. The research title is exploring the link between unsupportive parenting and social media release self-esteem. And the research advisor is Dr. Kara Winkler. So Bianca, is your plan for now?
Thank you. Okay, can everyone see my screen?
Yes.
Okay, hello everyone. My name is Bianca Mendoza, and welcome to my presentation, which explores the link between unsupportive parenting and social media related self-esteem. In the following slides, I will just discuss the importance of my study, explain my variables, share the trends and concepts found in the literature review, explain the origin of my research question, share methods and findings, and lastly, discuss the implications, limitations, and future directions for my study. So first, we have the importance of my study. My study is important because it looks at the relationship between two variables that have an influence on self-esteem. One of those variables is unsupportive parents, and the other variable is social media. So most existing studies on self-esteem only focus on one or the other variable. So what my study aimed to do was make a discovery about behaviors that parents use with their children and the influence that it has on social media related self-esteem. So now onto the variables. So like I just mentioned, my two variables are the unsupportive parents and social media. So what do I mean when I say the unsupportive parent? Well, this variable was measured using 3 items, which are listed here. The first one is, generally, my parents have been unsupportive of the things I like. 2, my parents have not been there for me in moments of personal success. And 3, my parents don't seem to approve of my passion. As for the second variable, social media, this variable measures how someone allows social media to affect how they feel about themselves. Now on to literature review. My study used or reviewed 4 scholarly sources pertaining to parenting styles. And one recurring trend that I found was that parents who were supportive and were active in their children's life had a positive effect on their children, and they had a healthy self-esteem. Another trend that I found was that parents who showed unsupportive parenting styles, such as authoritarian, neglecting, and rejecting behaviors towards their children, had children who had low self-esteem and a difficult temper. So next we have the literature review, which focused on social media and its influence on self-esteem. It also focused on other constructs such as mood and repetitive negative thinking. So one concept that I noticed from this literature was that different uses of social media resulted in different consequences on self-esteem. So for example, productivity on social media had a positive effect on mood and self-esteem, whereas using social media to vent or share feelings didn't result in these positive effects. Another example is that people who scored in a high social comparison orientation had a lower self-esteem. One finding from the literature that stood out to me was that most people don't claim their self-presentations on social media to be purposeful, but they do claim that other people are. So here's the origin of my research question. Parents are the first people that we interact with, and they play such a big role in the kind of people we grow up to be. So I reasoned that it would have an influence on self-esteem, susceptibility to social media, which led to the development of my question, which is, what is the relationship between the extent to which parents are unsupportive and how much social media affects how someone feels about themselves? So now we have the methods for participants. My study included a total of 39 participants, all of which who were students that were recruited from an undergraduate communication study course. The average age for participants was 28.61 years old. Over 3/4 of the participants identified as either cisgender man or woman, and the remaining identified as either agender female or chose not applicable or preferred not to say. For procedures, my study used a cross-sectional survey research design, which basically means that a survey was used to gather the information from the participants. And then we have our measures. Like I mentioned earlier, the unsupportive parent variable was measured on a 3-item mean, and each item was measured on a 1 minimum to 5 maximum scale. And the mean resulted in a 2.26, the standard deviation in a 1.13. And the second variable was social media affects how I feel about myself, which was only 1 item and also used that same scale, a 1 minimum to 5 maximum. And the mean resulted in a 2.59 and standard deviation in a 1.25. So for findings, the results indicated that there was a non-significant weak correlation between the variables, which is reflected here by the R and the p-value, which reflect the correlation significance value. The R resulted in a 0.13 and a p, and the p-value was greater than a 0.05. And now we have the implications, limitations, and future directions for the study. So the biggest limitation that I found in my study was age differences among participants. So meaning that the participants in my study, their average age was around 28 years old. The participants used in the scholarly sources related to parenting styles and their influence on self-esteem were all children under the age of 13, and the participants used in the scholarly sources pertaining to social media and the influence it has on self-esteem were all students in college as well. For implications, the results show that there was a non-significant relationship between the variables. However, they still have a strong impact and influence on self-esteem, whether that be a positive or negative impact. So for future directions, I would say to continue using the same variables and studying them, but consider switching the focus of the question or the direction of it. So for example, how do parenting styles influence an individual's use of social media? And I would like to thank Dr. Winkler for being my mentor and helping me through this journey. And that concludes my presentations, and I welcome any questions.
Yes, I have a question.
Yes.
Yeah.
Is one parenting style more effective than another? Like, is a mother's more positive or volatile than a father's?
What was that last part? I don't think I understood the last part.
Is a mother's parenting style more positive or negative than a father's?
My research didn't look at that specifically, but from what I found from the literature review, parents in general just differ in their styles.
Okay, good. Any other questions? Anyone?
Yes, I have a question.
Yes.
I'd like to know, how did you come up with the criterion that you presented in your study?
What do you mean by criteria?
So you talked about what qualifies a person. For example, you talked about what qualifies a person as feeling unsupported by their parents. How did you come up with these different qualifications?
Yes, so like I mentioned in my presentation, the study method design was a cross-sectional survey research. So my study was a part of a bigger study, and it was a part of the class. So other students incorporated these questions as part of their study.
Okay, good.
That makes sense. Thank you.
Thank you.
Great. Well, continue researching. That's an interesting topic and certainly timely,
Thank you.
as we hear that the internet or social media is negatively impacting at least mental health. So
thank you.
I don't know if that's proven, but they indicate that.
Thank you.
Okay, we'll move on to our second to the final oral presenter. Been wonderful morning of presentations. So we have Stephanie Davis and Rode Villarreal presenting on plastic recycling at the University of Houston downtown. Their research mentors are Mr. Juan Martinez, Dr. Lisa Morano, and Ms. Shoshren Hashimi.
Thank you so much for that warm welcome. My name is Stephanie Davis.
And my name is Rode Villarreal, and I would like to share a presentation with you.
We hope you liked our shirts. They're made from 100% recycled material, including plastic.
Recycling.
We're very excited to share our research on plastic recycling at the University of Houston downtown as part of a project for the Eco Representative sponsored by UHD's Center for Urban Agriculture and Sustainability.
Plastics are made from a natural resource, crude oil, that for the most part, we use it and throw it out. But did you know that recycled plastics can help create clothes, tennis shoes, dinnerware, reusable shopping bags, and even buildings? It can. Recycled plastics can go right back into the shelves in many forms. Recycling also helps with the economy by creating jobs, reusing natural resources, and keeping plastics out of the landfills and oceans, in turn helping biodiversity. Landfill trash releases pollutants like methane and carbon dioxide into the air, the soil, and drinking water. And in the landfills, plastic may take up to 1,000 years to decompose. This is a critical concern that affects all the living organisms.
Under the direction of Juan Martinez and Dr. Morano, we investigated how UHD conducts its plastic recycling and what we could do to encourage more recycling at the university. We began by asking what the plastic recycling process is at UHD and who or what departments are involved in this process. Then we wanted to know if there were any improvements that we could make to the recycling process.
After meeting with Mr. Juan Martinez, we met with department managers in charge of collecting and processing the recycled items. We researched the best recycling practices of other universities and brainstormed ideas on what projects would work best here at the university. We took inventory and weightings of the current number of recycled plastics on each floor of the university buildings. And we also spoke to students throughout the university, and you saw the data to put together our action plan.
So these were our findings. We found that there's an average of approximately 10 pounds of recycled plastics that are collected within campus and sent to the facilities department every Thursday. And those are then transported to UH main campus. We found that plastics that are being recycled properly with items like empty sports drinks, water bottles, soda bottles, as pictured there in the first picture, and improperly recycled items in the second picture with things like greasy napkins, leftover food items, like pizza crust, face masks, and cleaning wipes, things that do not belong in the recycling bin. We also found that active participation amongst key stakeholders in these departments is vital to the success of the recycling program.
As part of our action plan, I designed a digital slide that rotates on all the screen monitors of the university campuses, promoting the UHD community to reduce, reuse, and recycle. I created a website with information on how recycling is done here at the university and information on where else around the city of Houston the public can recycle. The website myparkrecycling.wordpress.com was entered into a national competition with Oak Ridge Institute for Science and Education, and they promote plastic recycling as well. It was a great experience and exposure to what we are doing here at UHD to help with plastic degradation in the landfills.
We also repositioned recycling bins that were in low traffic areas or hidden behind doors to more visible high traffic areas. We allocated a recycling cart for the UHD food market that's located on the second floor of the main building. With this additional signage in the works and more educational interactive student events, we want recycling to become a social phenomenon at UHD. The Center for Urban Agriculture and Sustainability is very excited about the future collaboration with Coca-Cola and UHD to help further promote plastic recycling on campus and within our Houston communities.
That's right. And in conclusion, we learned that plastic recycling greatly impacts the sustainability triangle. We are proud to say that UHD is contributing to the sustainability of our natural resources. And in working with various department leaders, we found that they are happy to work together in this endeavor. And that is key to making sure that the program is successful. We interacted with students and found that they are motivated to recycling. So that is also an awesome thing. The UHD and CUAS continues to provide opportunities for students to proactively make a difference. Gators encourage Gators to recycle. And together we make a difference.
If you'd like any information or have any questions about what we did specifically in the research program, please contact us on our emails or the Center for Urban Agriculture and Sustainability. And we want to note that this project was funded as part of an EcoReps program through the CUAS.
We would like to thank Dr. Morano and Juan Martinez for walking us through this process, and also Professor Hashemi for encouraging us, as well as the Scholars Academy for allowing us to be part of this awesome research conference. If you have any questions, we would be happy to answer them now.
So wonderful job on Stephanie and Rob. This is very meaningful. I just have a quick question. So now we are actually finishing the COVID now. We get a lot of waste from the COVID, like as you mentioned, the face mask and then those gloves. And do you think those things are recyclable? Of course, plastic is recyclable. Have you considered those?
No, we did find recycling in the recycling bins. We found face masks, and you could see cleaning wipes as well. Those were pretty common throughout our audits, but those, as far as we know right now, they're not recyclable.
Mitsu?
You know, like I remember the student was that the group that actually started on this recycling on campus many years ago, and but right now it seems like you know, the paper recycling bin in some of the labs and the plastic recycling bin outside of the labs, they look, all they're blues and they look a lot alike. So I think a lot of I find that I see some of the plastic in paper recycling bin and vice versa, that kind of thing. So I think the country that actually does a lot of good recycling, they typically have the 3 different recycling bins with the different colors, like maybe for the cans, plastic, and the papers. And I was wondering if we can shoot for something like that on campus, and you know, maybe that once people get used to doing that, maybe we can split to the community maybe. And I just want to commend you guys for really working hard and you know, to recycle, and I think it's a great thing. And I hope you guys continue, and you got my full support.
Thank you so much.
Thank you so much.
That's
Yes.
a great point as well that you made up, because that's something we noticed as well. In the COAS in the STEM building, the recycling bins are labeled, and it's very clear. However, in some of the on the floors in the academic and main building, they are different. There are green colors, there are blue colors, there are some that say plastic only. And so that is also some of the signage that we are working to create. So it's just pretty cut and dry. And it says, you know, soda bottles, plastic water bottles go here, because you make an excellent point. There are different recycling bins, and it can be confusing because we found students will recycle, but they just don't, you know, they're confused on, well, what do I do here? This one says paper only, and then this one is, you know, they're all blue, but then there's some green ones too. What does that mean? So we are working on that, and we have to, hopefully we'll get approved to get these things completed soon.
I think when everybody starts seeing that maybe 3 sets of different colors all over the place, I think, you know, a lot of people are visual. So even without reading or writing, they get used to putting stuff in a right.
That's right.
Yeah.
And that was some of the best practices at the other colleges and universities that we researched as well. They are doing that.
Well, you have a lot of questions in the chat, and I'm going to let you move to that. Great presentation.
Thank you.
And answer those questions. Thank you. Okay, Mian, if you would introduce our final speakers.
Yes, so our last presenters will be Sabrina Portilla and Jonas Zabri, and then the research title is Antibiotic Sensitivities of S. marsicans and E. coli bacteria in 2 modes of growth. the co-authors is Sabrina Pautina, and the research mentor is Dr. Poonam Gulati Sahotra. So Sabrina and Jonas, yeah, please share your screen.
Oh, let me get just one moment. There we go.
Can everybody see that?
Yes, we can.
My name is Yunus Saberi.
And I am Sabrina Portilla.
This is a presentation on antibiotic sensitivities of planktonic and biofilm cultures. Bacteria are found in two growth modes, planktonic and biofilm. Planktonic bacteria are free-floating individual bacteria that can be found in places such as running water. Biofilms are communities of bacteria that can form on damp surfaces. In nature, they're often found with more than one species of bacteria. They also produce extrapolymeric substances which surround the community and help them be more resistant to environmental stresses. Biofilms and planktonic cultures are also different because they express different genes.
Clinically, it is commonly known that microbes in planktonic states are less resistant to antibiotics than in biofilms. There are studies that suggest that the biofilms are more tolerant of antibiotics due to several factors such as slow growth, factors that make it harder for the antibiotic to reach the microbes, and possibly unknown resistant mechanisms. It is becoming a problem we're seeing more and more in infections that are formed in multi-species. The purpose of this study is to test the effects of the antibiotic kanamycin on mixed species cultures of E. coli and Serratia marcescens in planktonic and biofilm states. Materials in this experiment include the bacteria E. coli, Serratia marcescens, media, and other plasticware. They are cultured on trypaxoid agar plates and then grown in as a liquid culture to reach their log phase, where the bacteria are actively dividing. This is the most effective phase to use antibiotics. Kanamycin was used to perform the minimum inhibitory concentration and minimum bactericidal concentration in planktonic, while in biofilms, we use the crystal violet colorimetric assay in place of the MIC. We also do differential tests like eosin methylene blue and phenol red tests to distinguish bacteria. Minimum inhibitory concentration is the concentration at which the antibiotic stops the growth of bacteria. The first tip to the very left is our media, which make it our negative control. Then is our positive control of 0 micrograms per milliliter with no antibiotics added. So we set up the concentrations of antibiotic from 0 all the way through 1024 micrograms per milliliter. And starting on our second tube, the amount of E. coli and Serratia added are the same. As shown in our figure, our planktonic results for the mixed bacteria show growth in tubes with 0, 2, and 4 micrograms per milliliter, where the bacteria was not inhibited. The first clear tube shown here is 8 micrograms per milliliter, and that indicates our MIC.
Figure 2 shows the minimum bactericidal concentration, which is the lowest concentration at which the antibiotic has killed all the bacteria. As we go higher in the concentrations, the antibiotic's effects can be seen as the plates gradually show less and less bacterial growth. The first clear plate with no bacterial growth is 512 micrograms per milliliter, so our tests indicate that that's the MBC. Table 1 has data from single species planktonic MIC and MBC results, which show that both bacteria were inhibited and killed at 2 and 4 micrograms per milliliters respectively. This shows that even in planktonic form, the multi-species cultures are more resistant to the antibiotic. To test for biofilms, we conducted a colorimetric assay, which is used to check the integrity of the biofilms. This test works by adding the dye crystal violet as it attaches to the cells present. The sample is then put into the device called a photospectrometer or plate reader that emits a light through the sample. The light is absorbed by the dye and only a certain amount passes through. That is how we get the absorbance values. When the concentration of bacteria is high, less light will pass through. In this figure, we can see that at the earlier concentrations of the antibiotic, the bacteria still grows and has a drop at 16 micrograms per milliliters of antibiotic. After that, in the higher concentration, the bacterial growth stabilizes. When single species biofilms were tested, they showed results that were similar to the mixed species, where there was a drop at the lower concentration of antibiotics, and at the higher concentration, they start to stabilize.
We were predicting both Serratia and E. coli would either survive equally throughout all the concentrations, or that one species would dominate in the lower concentrations while the other dominated at higher. We played it to find out if the species are either helping each other grow or obstructing it. Preliminary data shows that Serratia dominates at low concentrations of antibiotic. This can be seen by the heavy red pigmentation in the plates 0 and 8 micrograms per milliliter that distinguishes Serratia colonies. Even without any antibiotics present, as shown in the first plate, Serratia is still dominant. The white colonies show more abundantly in the higher concentrations of 128 belonging to E. coli, and overall they seem to be helping each other's growth because as the concentration of antibiotic increases, the bacteria still survive. To conclude, our results from planktonic cultures show Serratia to be the only bacterium present throughout higher concentrations. However, both organisms, E. coli and Serratia, are shown to be present in lower concentrations of antibiotic.
We have also determined that in the mixed species planktonic, the MIC is micrograms per milliliter, and the MBC is 512 micrograms per milliliter. They're higher than the individual species cultures. This data suggests that mixed species planktonic infections would be harder to treat due to the interaction between the organisms. In preliminary data for biofilms, we see a drop in the concentration at 16 micrograms per milliliter, and the biofilms are growing at the higher concentrations. This would suggest that biofilms are susceptible to antibiotics at lower concentrations, and the antibiotics start to lose their effects when they're in a higher concentration. In addition, Serratia is surviving at the lower concentration of the antibiotic, whereas more E. coli survives at the higher concentration. These results suggest that antibiotic concentrations are changing the organisms which are dominant in the biofilms. We have also considered future directions for the project where we can test different stressors like bacterial viruses known as bacteriophages, or we can add another antibiotic and maybe add a different organism that would somehow change the interactions between the organisms.
Thank you, Jonas. And okay, go ahead.
Special thanks to Scholars Academy for allowing us to present at the SRC, Dr. Gulati, our research mentor, Mr. Medias Millian, Mr. Frank David for having supplies and support readily available. We also want to thank the Department of Natural Sciences for the new facilities and our lab mates, Annie and Anna, for all the support they've given us. Does anyone have any questions?
Excellent. Do we have any questions?
I have a question for these two great presenters. Have you considered the temperature effect for your study?
The temperature we usually go for is room temperature. It's around 30. It's room temperature for both the mixed species cultures, and individual temperatures, it's whatever the organisms grow optimum at. So for E. coli, it's around 37 degrees Celsius, whereas Serratia, it's 27 degrees Celsius.
Good.
Anyone else? Well, I think your study is so important. You know, when you consider even hospitalizations, the idea that where you have the combination of bacteria and some will be killed and some will not, you know, you can only have so many antibiotic, what do they call them? Where they combine them to, what is that called?
Cocktail.
Yes, thank you. Cocktail, you know, and then they adapt to that one or those combinations. So it's really a very interesting question. Okay, anybody else? Well, okay, thank you so much. And we're getting ready to leave for lunch. I just want to say thank you so much to all of the presenters. You must have practiced. You did a wonderful job staying in your time limit. And thank you to all the people present, undergrads and faculty and guests. You know, you really did a great job of presenting your data and having interesting topics. And I really congratulate the SRC planning committee members, because they were the ones that ranked and put together our oral presenters. So, and faculty can't say enough. You're the ones sponsoring this research, and just continue to do the wonderful job that you do. And undergrads, I would only say, be brave like Dr. Trong and go ask a professor to join their lab. And if you don't get a positive, ask another. Although here at UHD, many are pretty positive. And if they're filled with 10 or 12, you might get a negative, but continue trying. So we'll end this session. And remember, we'll come back into the campus groups SRC fair, and we'll start the poster presentations at, let's see, Mercedes, do you want to, I don't want to get it wrong, at 12:15. All right, so enjoy your lunch on your own, and we will see you then. Thank you so much.
Thank you, everyone. Thank you, Dr. Parker.
Thank you, Dr. Jang, for helping. Okay, and I have a question, just as you're leaving, I have a question about the PowerPoints. We will arrange to put those, and part of the recording as well, put those in our SA YouTube. All right?
The poster session says 12:05, Dr.
Yeah, but 12:15 is the session one, I believe. So we're going to take that 12:05. I don't think you need 10 minutes to get into the campus groups.
So just change it to 12:15.
12:15. All right. Thank you. I'm going to close this out now.
Bye, everybody. I think we had 181 major, that most we had.
Yes, I'm going to end this.